This is podcast number 13, and this is the last of our walk through the filaments that make up the cytoskeleton. So these are intermediate filaments, and they are not the smallest. They're not the biggest diameter. The biggest diameter would be microtubules. Smallest diameter would be microfilaments or actin fibers. They're composed of lots of different kinds of proteins, not one family. So in microtubules are made up of tubulins, alpha, beta, gamma tubulins. Microfilaments are made up of actin, G-actin, and intermediate filaments are made up of all kinds of stuff. These intermediate filaments are not polar. So again, microtubules, you have a plus N and a minus N. Same for microfilaments. And the plus N is the N that is actively growing or more actively growing, and the minus N is less actively growing or actively shrinking. Intermediate filaments, that's not the case. Also different is that these are much more stable. So when they're laid down, they tend to stay in that conformation. They can be reworked as the cell grows and as the cell changes shape, but they are much more stable minute to minute than a microtubule or microfilament would be. And they are much more cell type specific. So when you get into working in a lab, there is often a particular protein you're interested in because it pertains to some phenomenon you're studying, if it's cancer or heart disease or cold acclimation in fish or whatever. And if you want to see if that protein has gone up or down as a result of something you did to those cells, you have to express that per some kind of background protein. So per the amount of protein that we always expect to be in cells, has this particular protein changed or not? Those background proteins are called housekeeping proteins, and the most common ones that are used are tubulin and actin. There are some other ones, but the point is that actins and tubulins are in all kinds of cells. And so no matter what kind of cell is out there, you can be sure that they're making tubulin or actin. That's not true for intermediate filaments. The type of cell will have a different type of intermediate filament depending on what type of cell it is. And in the case of metastatic cancer, this is cancer that started growing in one part of the body, became mobile, and moved to another part of the body. When it's detected in that other part of the body, it can be hard to figure out where did this originate. So what kind of cancer was this originally? And you can use the kinds of intermediate proteins that are in that cancer as a clue as to where that cancer started. So did this start in the liver? Did it start in the lung? Did it start in the bone marrow? That sort of thing. And intermediate filaments can tell you that. Okay, it's just pictures of intermediate filaments. You've seen a bunch of squiggly lines on these micrographs. These are different kinds of squiggly lines, and the filaments are intermediate in size between the thinnest filaments, microfilaments, and the thickest filaments, microtubules. So I said there's a bunch of types of intermediate filaments, proteins out there. Just mentioned some of these on this chart. So keratins are a very, very common intermediate filament. Keratins are proteins that make up filaments in your skin and in your hair. And in proteomics, which I used to do a lot of, don't do so much anymore, the most common contaminating protein that you would find on your gel would be a keratin, because the people doing the proteomics are humans, and they have intermediate filaments all over them on their hair and skin, and those tend to fall into the analysis And so the most common protein you would detect would be keratin. There are vimentins, desmins, GFA proteins. These are typical to different kinds of cells. Vimentins in fibroblasts, desmins in muscle cells, GFAs in glial cells, and astrocytes part of your central nervous system. There are neurofilament proteins that are typical to central and peripheral nerves. And then there are nuclear lamins. Later on we'll talk about how nuclear lamins can polymerize to help form the structure of the nucleus after mitosis, and depolymerize during prophase when the cell is getting ready to go through mitosis. So this is a protein network, meshwork that keeps the nucleus in a particular shape, and those are called lamins. An example of how you get from a protein to a filament in terms of intermediate filaments, you start off with a dimer, so two copies of the protein that intertwine with each other. These dimers can then form a tetramer, two dimers coming together to form a tetramer. And then these tetramers can assemble end to end to form a protofilament on its way to becoming a filament, and then protofilaments assemble into an intermediate filament, like so. So your hair, for example, at a molecular level would have a similar sort of structure to this. Intermediate filaments can form connections and cross bridges between other kinds of cytoskeletal elements. So here's an example of a false color picture of the cytoskeletal network of a cell, where the microtubules are the large diameter fibers labeled in blue. And then you have different kinds of intermediate fibers forming connections among these microtubules. And those are labeled with different colors. So it's not that microtubules stayed in themselves and actin fibers stayed in themselves and intermediate fibers stayed in themselves. They're all interconnected to form that soggy nerf ball that is the cytoskeleton. And then one last H5B question for you.